Good morning all. Myself is Mrs. Ghodake-Patil, Swami Ramanand Bharati Junior College, Tasgaon. Today we are going to continue with the new point from the reproduction in lower and higher animals. That point is development of embryo or embryonic development. Actually, the zygote formed by the syngamy is automatically divided, and that divisions studied as first cleavage, then that cleavage continues into the blastulation, then blastulation continues into the next step, that is implantation, and implantation finally forms or develops or continues into the gastrulation. So these four stages continue with the embryonic development, and in that step the embryo forms. So while studying this process, the zygote which is diploid formed during the formation of syngamy or with the syngamy undergoes divisions. That first is, now we continue with the cleavage. In the cleavage, there are number of sequential divisions. That means actually cleavage is nothing but it is the early mitotic division. We already studied in the last time, that is the zygote forms or zygote undergoes, or during the gametogenesis different processes takes place. There are mitotic divisions, then meiotic divisions. So such mitotic divisions are slightly different here. Actually cleavage is formation of the blastula or blastocyst. That blastula forms by these stages. It doesn't shows the growth phase like observed in the mitotic division. There is equal division, but the cells which are formed in the mitosis during growth phase are reduced in size. See here, the first cell undergoes division. It shows two-celled structure, and in that two-celled structure, one cell is somewhat larger than the other. It also shows polar bodies. The cells which are formed are known as blastomeres. That blastomeres are small in size. As the cells gets reduced, it has high metabolic rate. It also shows the more replication of DNA and more oxygen consumption. Then we continue with the process of cleavage. The cleavage in human being, the cleavage is holoplastic. That means whole zygote gets divided. Here there are different stages of the formation of the cells, that is longitudinal or equatorial, longitudinal or meridional, and horizontal or equatorial. So the first stage of the cleavage formation, that is two-cell stage, in which the first cell undergoes division after 30 hours of the fertilization. It forms two cells, one is smaller, another one is somewhat larger. It also shows polar body. Then the second cleavage also shows the divisions of longitudinal divisions, and it forms the right angles to the other. It also takes place the 30 hours after the fertilization. Then third cleavage, which forms again division, that means in mitotic, first cell or single cell form 2, 2 cell form 4, 4 cell form 8, 8 16, 16 32. Like these cleavages takes place. Then here the covering of zona pellucida is present. Then this 4-cell stage gets converted into the third cleavage, that is 8-cell stage. Here thus at central this 8-cell stage continues into the fourth cleavage, that is 16 up to 32. This 16-cell structure is like the mulberry. तुतीच्या झाडाची फळं असतात त्याप्रमाणे ते मलबेरी लाईक स्ट्रक्चर दिसतं. त्याच्यानंतर हे मोरुल दॅट इज नोन ॲज मोरुला. द मलबेरी लाईक स्ट्रक्चर इज नोन ॲज मोरुला. It is 16 cell stage. Thus the number of different cleavages takes place. Now see here, the central cells or inner cells are larger and outer cells are smaller in size. Then this process continues into the next stage. This morula form, this zona pellucida is present, appears there. The size of the embryo remains as it is as the zona pellucida is present. Only the division of the cells takes place. Then it continues into the next